Welcome, everybody. And today we will have I think, great event together. And we have some open campaigns. And I'm so excited to show them to you because each time when I work with them, I am so happy to help their future because I can see the potential impact from all of them. So my name is Damla. I'm a communications director at Capital Cell. And today we will do presentations, 8 minutes presentations, and after we will go to the breakout rooms to ask our questions to founders and CEOs. So please wait until that time so you can ask your question who you want. So maybe we can start with Renaud.
Thank you very much, Damla and Elita, for setting up this meeting. My name is Renaud. I am the CEO and co-founder of Alaya.bio. Alaya.bio is a biotech company that I created in 2022 together with my partner in crime for the past 15 years, Cécile Bosch, the CSO of the company, and Michel Sutherland, our scientific co-founder, and who is one of the CAR T pioneer and most likely a Nobel Prize winner in the next few years for his work on CAR T cells, which is the topic of today. We have a team of 20 people based in Paris, plus an additional 3 people as part of the collaboration that we have with Michel. So Alaya is developing therapies to fight against cancer, and more specifically, we are developing immunotherapies. Unlike chemotherapies or unlike radiotherapies, which are targeting directly the cancer cells, immunotherapies will stimulate the immune system of the patient to allow him to fight against cancer or to fight against autoimmune diseases. More specifically, over the past few years, a new technology has revolutionized the treatment of some cancer, and more specifically blood cancer. And this technology is called CAR T therapies. So what is CAR T therapies and how does it work? So you have here a little schematic on the slide. And basically, for CAR T, you have to take a blood sample from the patient. You ship it to a centralized lab where you isolate the T cell, the white blood cell, or the cytotoxic cells. And once those cells are isolated, you introduce a genetic modification to force the expression of a specific receptor specific to the cancer cells that you want to track and kill. Once this genetic modification is inserted, you force the proliferation of those cells to build a little army that will be reinfused to the patient. You are on the right part of the slide, a little cartoon that is basically presenting what CAR T is. It's basically creating some super soldiers. And the red piece that you can see on the right hand is basically the CAR T and the genetic modification that is inserted into the CAR T cell. So it works pretty well with outstanding clinical results, and more than 7 CAR T therapies that have been approved to fight cancer. But as you can hear, it took me almost a minute to describe you in length how CAR T works. And what you can also understand between the lines is that behind this complexity, there is obviously a consequence in terms of cost of this kind of treatment, because this kind of treatment will cost approximately $500,000 per patient. At Alaya, we are developing a targeting nanoparticle to produce the CAR T cell directly in vivo or in situ. And the objective is to produce faster, cheaper, but also better T cell. Faster, it's because we can replace a weeks-long manufacturing process by one systemic off-the-shelf administration of the targeting nanoparticle. Cheaper because you don't need the costly manufacturing facilities to produce ex vivo the CAR T cell or to manufacture the CAR T cell ex vivo. You have also a shorter and simplified supply chain, and better T cell because the cells that will be produced in vivo are de novo cells They are not exhausted by weeks of expansion or weeks of overstimulation of the cells, and they are fresh cells that are able to track and kill the cancer cells. What is also interesting to note is that this in vivo strategy allow, and this is unique to our technology, but allows the redosing potential of the patient if the patient relapse or if the patient needs a maintenance treatment. So how does it work? Without entering into too many details, you have here a schematic of the targeting nanoparticle. In the middle, in light gray, you have the lentiviral vector. So the vector that is coding for the CAR, basically the same than the one that you use ex vivo to produce ex vivo the CAR T cell. But this vector is further functionalized with some biopolymers, the red and the blue piece that you can see on this drawing, onto which a targeting agent is covalently grafting. And this targeting agent will basically act as a zip code to redirect, once injected into the bloodstream of the patient, to redirect the targeting nanoparticle toward the cells of interest, in this case, toward the T cells. So as mentioned, we have an active collaboration with Michel Sadolin, one of the CAR T pioneer, and we started this collaboration when he was at Memorial Sloan Kettering in New York, and we are continuing the same kind of collaboration at Columbia, where he recently moved. Michel is responsible for the translational collaboration, i.e., the generation of all the efficacy and safety data that will be required to move to clinics. We recently passed a very important milestone with the completion of a first preclinical package that allowed us to designate what will become our clinical candidate. So basically, the formulation of the nanoparticle that will be used in clinic. So this, what has been recently achieved as part of the collaboration with Michel, and the objective of the next coming month will be to put in place a first clinical trial to evaluate this targeting nanoparticle in patient, and more specifically, we plan to put in place an IIT, an investigator-initiated trial in China, where we will be able to treat about 20 patients, with the first patient treated before the end of next year. So the objective of this IIT is to get the confirmation of the safety, but also to gain the first evidences of the clinical efficacy of our in vivo CAR T platform. As mentioned, about 20 patients, and with this first construct that we have elaborated. What is interesting is that the in vivo field is obviously booming. As you may have read recently, no less than 4 acquisitions took place over the past 6 months, with 3 of them with a value beyond the $1 billion for the acquisition. And what is also very interesting, as you can read here, is that the stage of development where those acquisitions took place are pretty close to the one where we are currently at Alaya, because Biotech, for instance, got acquired by AstraZeneca for $1 billion, and they only had 1 patient treated. Likewise for Orbital, which was recently acquired for $1.5 billion, only based on IND-enabling studies, i.e. preclinical data. So based on this obviously very dynamic environment, M&A environment, but also on those promising preclinical data that we recently obtained, we signed a term sheet with new investor for $10 million, which pushed us to recently announce the closing of the crowdfunding campaign with Capital Cell. So you only have a few days left. I don't know if it is on Thursday at midnight or Friday morning, but only 1 or 2 days to join this venture together with us and to be part of the future of in vivo CAR T.
Thank you so much. And the campaign will close on Thursday midnight. So they have time to invest and don't have that much time to invest. Yes, thank you so much.
My name is Fabian, and I happily present autonomyo to you. Autonomyo is about empowering neurorehabilitation along the whole continuum of care, which means not only in the post or the acute phase where most solutions tackle, but along the way a patient goes through. And that's what we stand for. We want to bring back mobility to people who have lost it. So we are in the space of rehabilitation, stroke, MS, neuromuscular dystrophy, etc. And I share a quick story so you understand where the problem today is and what we try to tackle in that space. This is Sarah. She's 58 years old and she had a stroke last year in May. She had a blood clot that was removed in an acute hospital in the emergency department. That was quite fast, but she still had severe disability and she was not able to move the left part of her body. So lower extremity and upper extremity, she was not able to move. She got into rehabilitation after acute hospital for about 3 months, so quite intense. There she had many training sessions. It's 1 or twice a day for physical, and then you get other therapies on top of that. And she could, by the end of those 3 months, walk with a walker, with a help to go through the basic mobility steps. At home, she had once a week physiotherapists in an outpatient center, and once a week someone came to her home, or still does, and she gets a treatment. Unfortunately, she doesn't do any progress anymore. So she reached a plateau because those 2 sessions a week are way too little, and she's not able to really train on her own. So that happens to many patients, that they reach a plateau way too early because they lose the intensity of rehabilitation, the intensity of training, because the follow-up treatment after the inpatient setting is way too little. Lack of therapists is a big reason, lack of training devices or robotics that would assist that, too far travel distances, etc. And in this setting, we built tech to enable training without the need of constantly having a supervisor or a therapist with you, and to get the best accessibility to your rehabilitation as possible. Currently, we have 2 products in the pipeline. The first is Skywalk. It's a very simple tool that prevents patients like Sarah from falling, to be able to verticalize and to do simple training steps, side steps, little squat movements, etc., to really train. It's adjustable, it's affordable, and it allows a training that Sarah would not be able to do otherwise. In a clinic, in an outpatient center, but possibly also at Sarah at home. For example, that treadmill setup that you see here, bottom right, a test patient of us was having that setup at home and could do daily training sessions with that after someone helped in the Skywalk to start with. So that bodyweight support system is for all kind of neurological patients, tested and suitable for them. It's a class 1 medical device, and we plan to launch that beginning of next year, first in the US and then in Europe in Q2. The pre-series production is currently in a last clinical test in Zug in Switzerland, and then the serial production begins. Our second product is a robotic device, the NeuroExo, an exoskeleton for people to train. Focus is stroke people, so it is adapted to that very specific need that oftentimes you have only one side that you need to train, and also the way it is designed, that it is able to work also seamlessly and as easy as possible in an outpatient center, and finally, eventually, potentially also in people's home once there is reimbursement for such systems. Switzerland has just introduced reimbursement for that kind of robotic devices in the outpatient fields, and other countries are working on such reimbursement systems. It comes with games, it comes with remote platform for a physio also to accompany a patient on that journey, and as a whole rehabilitation platform. World Health Organization reports it's about 25 million people that require neurological care and that kind of rehabilitation. And that's the group we built that kind of tech for. With me, there is Renaud in our team. Renaud is a senior executive. He had and he led 2 rehabilitation companies in the past for around 20 years, and joined our project around 1 year ago. And Amalric built the tech. He's from EPFL, and he's the R&D brain of the company. And together we launch those products. As said, Skywalk quite soon. The NeuroExo will take another about 2 years to really be launched in the market. And the current capital sale campaign is to fund all the market introduction efforts of Skywalk. And happily have your questions afterwards in the breakout sessions. Thanks a lot.
Now I want to continue with Emmett. Emmett, are you available?
Absolutely, here I am.
Okay, perfect.
My name is Emmett-Phil Coriat. I'm the CEO of FreeOx Biotech, a Barcelona-based biopharma company, and we are developing the missing piece in stroke treatment. My background is I'm Canadian. I've been in the biopharma industry for about 30 years, had multiple exits, and recently moved to Spain in order to be able to lead this company. Which has a very unique value proposition, and I'll explain to you why in the next couple of minutes. So stroke is a disease, but it is also an event. Lives are, turned around. Fabian spoke about stroke and no stroke very well. It's not really a disease. It happens in a few minutes. When you have a stroke, up to 2 million neurons die per minute. There are 13 million of them a year, and 1 in 4 of us will have a stroke in our lifetime. It means that there's a stroke every 3 seconds, and the survivors of stroke are left with permanent disability in about 50% of cases when it's a large vessel occlusion, which is the most devastating type of stroke that we are addressing at FreeOx. And what is really surprising is there hasn't been a new drug for stroke in 30 years. So that drug was launched in 1995 by Genentech, and it is a drug that basically dissolves the clot in the artery between the heart and the brain, which is called recanalization. In 2015, there was another advance in stroke called mechanical thrombectomy, which is a minimally invasive procedure, a little bit like when they put a stent in your heart, but in the artery between your heart and your brain. And that improves significantly the outcomes for patients, which means that the patients can live independently, carry out their daily tasks. But despite the event of mechanical thrombectomy, which basically removes the clot from the artery, the medical community was really surprised to discover that 50% of the patients are still left with disability and can't carry out their daily lives. And the reason is that there is something that they haven't looked at, which is the microcirculation in the brain, the smaller vessels in the brain that actually clot up, not clot up, but close up. And there's something going on there that explains why every other patient will still be disabled. What we have developed at FreeOx is focused on microcirculation, and it's reperfusing this microcirculation and is synergistic with the existing drug and mechanical thrombectomy. In 1995, this project, this drug phase 3 trial, last trial to the market, was funded by the NIH, and we have been selected by the NIH to fund our phase 3 trial. So we're in the same situation. I won't spend too much time on the mechanism of action, but basically, we are a cerebroprotectant. We have multiple targets, which are both inside the blood-brain barrier, but also outside the blood-brain barrier in the endothelial cells, basically the vessels at the cusp between at the blood-brain barrier. So we're the world's first cerebroprotectant. We are synergistic with current emergency treatments, be they the thrombolytics or mechanical thrombectomy. And we've demonstrated some strong clinical results, which I'll show to you in a few minutes. And we also have an important feature is that we have a 24-hour treatment window, which largely limits the use of the current standard of care drug, which has only a 4-hour window. In an emergency setting, this is a huge advantage. We have strong IP that goes beyond 2045, and our intervention, which is a simple IV injection, is easily and seamlessly integrated in the patient flow. We've demonstrated on a 421 patient phase 2b trial a 19% effect size, which means that 19% more patients with our drug versus placebo could carry out their daily tasks and live independently, not needing nursing home care or anything like that. So a huge saving to society and, of course, an improvement for patients' lives. Our drug has an excellent safety profile. It's derived from an endogenous molecule, and it has no increased hemorrhagic risk, so we can give it early on in the emergency room. What's unique about our value proposition is that we have been evaluated against 4 big pharma drugs for 5 years. There was a 5-year evaluation in the largest ever translational trial for stroke. And we came out on top, and the only drug that demonstrated clear clinical efficacy in a statistically significant way. As a result, we have a 30 to 35 million non-diluted financing from the NIH for a phase 2b trial. A phase 2/3 trial that is starting at the end of 2026. It's an 800-patient trial that is targeting a 9.5% effect size, which in effect is half of the one you saw before. So the target is quite reasonable, and the likelihood of success is very high. With regards to our fundraising, we're currently raising a 2.5 million Series A that has been fully subscribed via Capital Cell. We just secured it last week, but we're continuing the campaign for another few weeks. And the objective, the use of proceeds, is to prepare for this phase 2b trial, phase 2b/3 trial called Aurora. The minimum investment is 3,000 euros. And as a summary, why invest in FreeOx? It's a massive medical need. There's been no drug in 30 years. The burden of disease is massive, number one cause in disability. We are synergistic. We're not competing with existing treatments. It's a de-risked late-stage asset. We've got strong phase 2b data. We have a huge lever on the capital that we're raising because of the 30 to 35 million non-diluted financing from the NIH. We're addressing an underserved, addressable market. It's a 24-hour window for the treatment in the emergency setting. We have an experienced international team, a seamless integration into clinical emergency, and we have strategic momentum towards exit. We need basically 15 million to get to an exit. And as a result of this non-diluted funding and the results we have so far, we're expecting an ROI of around 19x.
Carlos, are you ready?
Sure. Okay, so as the other CEOs said, we are in the rehabilitation field, but not only in neurological conditions, but in chronic pain, neurological conditions, and we are working on develop cardiologic and pulmonary conditions. The problem that we solve is that more than 2.4 billion people don't receive the proper rehabilitation, and that problem leads to a chronification of the pathology and the decrease in their quality of life. This problem is due to limited access to rehabilitation, mostly for a shortage of professionals and a lack of effective therapeutic tools. The solution that we have found it's a combination between virtual reality and artificial intelligence. We have developed a solution that automates rehabilitation through autonomous sessions. We have developed programs that progress based on the evolution of the patient, increase the capacity of the physiotherapists or occupational therapists to treat more patients in the same time, and allows a reduction in the rehabilitation time, mostly for chronic pain patients, and improve the rehabilitation in the neurological patients. It's not only virtual reality and artificial intelligence, but it's integrated with a web app assisted app that where the professionals can prescribe the therapy and get data and collect data to improve the therapy. The use cases that we have working on now, it's are musculoskeletal and chronic pain rehabilitation, after stroke, post-surgical rehabilitation, for intensive care patients, and reducing anxiety in pediatrics and patients during invasive procedures. We have already demonstrated a 35% reduction in rehabilitation times and a saving for the extractors of around 800 euros, and the capacity of treatment up to 4 times more patients for each extractor. And it's important to understand that the virtual reality, it's a tool to improve the rehabilitation and reduce the rehabilitation, because it cheats your brain. For chronic pain patients, virtual reality, I don't know if you know that chronic pain patients have a lot of psychosocial factors related with the pain, and with virtual reality we can change the environment and the, let's say, virtual body, and that allows the patient to rehabilitate in less time. And for neurological patients with virtual reality, what we are allowing is to treat with the proper volume of rehabilitation and at the right time, because there is a window of around 5 or 6 days that if you start the rehabilitation before and with the proper volume, the prospect of the patient, it's quite better. We are targeting the European and United States market, and our principal differentiation is the ability to automate the rehabilitation through these programs that I have talked. We have a SaaS business model, and the pricing, it depends on the numbers of sessions that the structure have a year. We have already sell our product in South America, Spain, and Italy. We collaborate with many partners as Universidad de Granada, Sociedad Española del Dolor, many universities and distributors. And we have clients such as Clinic Hospital in Barcelona, Umibale Activa, or some hospitals such as San Joan de Déu Hospital. We are now around 16 people, and we collaborate with one of the best doctors in Europe that research in virtual reality and chronic pain. For now, we have we are growing year by year, and this year we will get around 200K of revenue, but we don't have still a proper sales and marketing team, and that's why we are raising 1.3 million to create this sales and marketing team. From this 1.3 million, we have already raised 1.15 million, and we want to close this round through capital selling at the end of December or the beginning of January. Thank you so much.
Thank you so much, Carlos. And now we are going to our last speech. So after Elita's presentation, we will go to breakout rooms and you can ask your questions to our CEOs. Let's continue with Elita. Welcome.
Good afternoon, everyone, and I'm Elita Montanari, the CTO and acting CEO of EVis Bioscience, which is an ETH spin-off that was incorporated in June 2024. So we are a young company. EVis Bioscience is based near Zurich in the Biotech campus in Schlieren. So RNA-based drugs might have a tremendous therapeutic impact. RNAs might be used for several medical applications that do not have a therapy yet. For example, they could be exploited for protein replacement, as vaccines, for cancer immunotherapy, for cellular reprogramming, and for gene editing. RNAs and DNAs need a carrier to be delivered and protected from degradation into our bodies. So far, only 2 delivery strategies have been approved by the FDA. Synthetic lipid nanoparticles that are delivering messenger RNA that we can find in the anti-COVID-19 vaccines from Moderna and Pfizer, and viral particles for the delivery of DNA in the nucleus of cells. However, lipid nanoparticles cannot target specific cells or tissues and can be toxic after repeated injections. On the other hand, viral vectors are immunogenic and have low packaging capacity. So this means that big genes cannot be packed into viral vectors. To overcome these limits, extracellular vesicles, shortly EVs, have been investigated for 20 years also in gene therapy. EVs are biological carriers that naturally deliver RNAs and proteins into our bodies. And depending on the selected EV, a different cell or tissue can be targeted. And this is very important for gene therapy because we want to deliver the RNA where is needed. So the main advantages of EVs are the biocompatibility because they are coming from our body and the targeting capability. However, they also face relevant limits, such as the scalability of the isolation and purification processes of EVs and the loading of EVs with exogenous and therapeutic RNAs without destroying the vesicle structure. So EVis Bioscience is the first company to employ a unique approach to quickly and efficiently load RNAs into EVs, forming these hybrid extracellular vesicles, shortly hybrids, offering novel opportunities for an extrahepatic delivery. So it means for targeting tissues that are out of our liver. Here we can see the morphology of hybrids derived from 3 different sources from the human body. So red blood cells EVs, mesenchymal stem cell EVs, and plasma. And we can see how the LNV is which is carrying the RNA, is hemifused to the EV surface. These carriers can provide specific targeting depending on the selected EV. More than 90% of messenger RNA can be loaded into our hybrids, and the hybrids loaded with messenger RNAs can transfect cells and express the protein of interest. Further, hybrids loaded with short interfering RNAs can silence genes in cells. So the EVis Bioscience business strategy is very versatile and mainly relies on three pillars and maximize both scientific and social benefit. The development and commercialization of the EVis nanovector to university and research institutions to support their research programs in the EV field that will be ready by mid-2026. The development of the hybrid carrier that can be licensed to pharma companies to expand their therapeutic pipeline, and the development of a proprietary therapeutic pipeline for treating rare genetic disorders. So between 2022 and 2025, big pharma companies, including Novartis, Pfizer, and AstraZeneca, acquired smaller companies developing gene-based therapeutics, almost all in a preclinical development, offering upfront payments of hundreds of millions. Two patent applications protect our technology. The first patent was filed in September 2023 and protects the chemical composition of LNVs and the hybrids. The second patent application was filed in February 2025 and extends the protection of the chemical composition of the hybrids and their medical applications. So EVis already won the Venture Kick phase 1 and phase 2, the Innovation Check, and just a few days ago, we also won the InnoSwiss with implementation partner, which is a competitive and very important Swiss grant. And this grant will financially support the preclinical research until 2027 and will allow the EVis team to expand. From mid-2026, the European pre-accelerator and the chemical kits with the sub-licenses will drive EVis to the 2027 when we will start to prepare the clinical batch, and then the InnoSwiss starting grant and eventually Venture Kick will carry EVis until 2028 when we are considering to make an exit after an IND or CTA. So the budget obtained will allow in 2020, the amount of money that needed by the EVis Bioscience to achieve the IND or the CDA is almost 12 millions, and planned inflows exceed these by almost 1 million. Our goal with Capital Cell is to raise 700 euros, and which will be used primarily to conduct preclinical investigations and to develop and commercialize the EVIT platform. And in 3 weeks, we already secured more than 70%, and the campaign will continue for more than a month. So significantly, the company's potential value could increase approximately 25 times by 2028. So the management team combines scientific, business, and financial expertise. EVis is further supported by a panel of experts in the pharma and clinical fields, and the CEO with a deep pharma experience will be hired by 2026. So the EVis technology already received financial support from several Swiss and European grants, and the company has ongoing scientific collaborations with international universities and experts worldwide. So if you would like to have more information about EVis, just follow us through the website, the LinkedIn page, and you can text us to our email. And thank you very much for your kind attention.
Thank you so much.